Hello students. Now let's start with the next enteric pathogen, campylobacter. Objectives: classification and biochemical characters, antigenic structure, viability characteristic. Now, campylobacter. These organisms were originally recovered from infectious abortions in cattle by Theobald Smith, in 1918, and they named it Vibrio fetus, as they were thought to be vibrios. Then they now placed this new genus Campylobacter in 1963. Understood? Campylobacter is a genus of gram-negative bacteria, typically appear comma or S-shaped and are motile. Okay? These are strictly microaerophilic. Gram-negative curved rods are motile, usually by a single flagellum at one or both poles. Okay? Now, Campylobacter species can infect humans, sometimes causing campylobacteriosis or diarrheal disease in human. Understood? Campylobacteriosis is usually self-eliminating and antimicrobial treatment is often not required, except when there are some severe cases or when patients are immunocompromised like in HIV. That time they need treatment. The most known source for campylobacter is poultry. As I told you, first it was found where? In cattle. Okay? But due to their diverse natural reservoir, campylobacter species can also be transmitted via water. Okay? Other known sources of campylobacter infections include food products such as unpasteurized milk and contaminated fresh products. Sometimes the source of infection can be direct contact with infected animals, which often carry campylobacter asymptomatically. We have already seen its morphological characters. They are curved, comma-shaped, rod, motile, and move via unipolar or bipolar flagella. They grow best in 37 to 42 degrees centigrade in a microaerophilic environment. Okay? They are also thermophilic. You have seen the temperature 37 to 42. They are thermophilic. Okay? And capnophilic also. I'm sorry, I have not mentioned it over here. Capnophilic means they require 10% CO2 for their growth. Okay? When exposed to atmospheric oxygen, Campylobacter jejuni is able to change into a coccal form. It can change its form. Understood? The number of known quinolone resistant Campylobacter strains is growing. It is suggested that this is caused by the overuse of quinolone antibiotics in animal agriculture. Let's see its cultural characters. Campylobacter is one of the 4 key global causes of diarrheal diseases. It is considered to be the most common bacterial cause of human gastroenteritis. Infections are mild but can be fatal among very young, elderly, or immunocompromised, immunosuppressed individuals. Species can be killed by heat and thoroughly cooking food. Now, to prevent infections, make sure to follow basic food hygiene practices when preparing food. Campylobacter species veterinary pathogens are C. coli, C. jejuni, C. lari. Extraintestinal pathogens, C. fetus. Abscesses causing pathogens, C. sputorum, C. concycus. Okay. This is the biochemical reaction. I am so sorry I have put this image, but if it is not clear, if you have any problem, if you are not understanding, please ask me. Okay. Now here it is shown species, catalase production, growth in 1% glycine, production of H2S. Okay. You take a pause, zoom in and just see. If still there is a problem, just let me know. Moving ahead, antigenic structure. Now, campylobacter, see, this is the structure of C. jejuni. Here you can see surface polysaccharide, flagellar modifications. Let's see. Campylobacters possess somatic O and flagellar H antigens. O antigen. Now, there's a scheme, Penner serogrouping scheme. 65 serogroups of C. jejuni and C. coli were made on the basis of heat-stable lipopolysaccharide O antigen. Understood? This is heat-stable lipopolysaccharide. Now next will be heat-labile HL surface and H antigens. On the basis of these two antigens, Layer serogrouping scheme 100 serogroups C. jejuni, C. coli, C. lari has been made. Plasmids have been detected in 19 to 32% isolates of C. jejuni and C. coli. Now let's understand cytotoxins, enterotoxins. Most strains of C. jejuni and C. coli produce at least two cytotoxins, one enterotoxin and two cytotoxins. The enterotoxin is similar to cholera toxin and heat labile E. coli enterotoxin LT and is neutralized by antisera to these toxins. No clear association between production of enterotoxin and serogroups or biotypes has been observed. Okay. Viability characters, those which are told about their survival, where they thrive, and their characters. Okay. In the next lecture, we will see its pathogenesis, symptoms, lab diagnosis, prophylaxis, and treatment. Thank you.